This technology can really do everything. We're able to move fluids, we're able to move particles within the fluids, change the temperature, split them, we're able to even make them explode. Basically everything you ever need to do in biology. The technology is a way to manipulate acoustic waves on surfaces and their interactions with liquids through these holographic structures. We call them phononic structures. The idea is that the structure is responsive to the acoustic excitation. So if I change the frequency, so the tone of the acoustics, I get a different shape of the waves on the surface. And so I get a different function. And so instead of having different machines that do different things at different time, I simply have to switch the frequency. And that's a button. And so all these different functions need different acoustic, we call them landscapes, but basically how the waves propagate on the surface. And that's the core of the technology. How do you control how to do that? And so what we do is we embed into this surface some macrostructures. And these microstructures interact with the acoustics and the waves and shape how it propagates. It's a technology that is so versatile. You can do everything with just that simple piece of technology. And one thing that in some ways is slightly different from other technologies, when you press a button, something happens. And it happens like that. It's very much developed to the point where we've got these kind of function bundles. So we know if we want to do movement of samples on a surface, what it is that we need to do. So then what people need to do to use it or make it available for their assay is simply change the reagents that they're putting in. The manufacturing processes are very well established, mass manufacturing techniques, and then putting things together is an engineering job that any product development engineering department would have.